Well, thank you very much, and thank you for having me. It's quite an honor to be here and to be invited to be on the board, of a place with so much history and with so much advancements, and have been made. And I guess my talk is a little bit different in the sense that it's not exactly about vitamin C, but it's about this concept, the concept of not killing the tumor, but actually making the tumor become a healthy tissue again. It's a concept which was actually pioneered at the center, and the concept of the healing as a therapy. And it'll become more obvious what I'm talking about as we go through the slides. Before now, let me say that there has been a revolution in the way that we, as biotechnology people and as the pharmaceutical industry, see cancer and cancer research. And that revolution has been centered around this concept of the cancer stem cell. So the title of my talk is, it's time now to actually strike the cancer at the root. So we're going to talk about stem cells a little bit, and then what is a cancer stem cell, why the war on cancer has failed. Now this, I guess, is a bit of a sensationalistic title. I want to get your attention. The differentiation is a cure. Supportive data. So we're not just going to be talking about some interesting energy theories here. We're going to have some actual data. And Dr. Reardon actually showed me a little bit before today some more exciting data, which you will hear about later tonight. And the next steps. So, what is a stem cell? Let's just go over the very basics. So, the definition of a stem cell is it's a cell that can make copies of itself, and then under certain conditions, it can become other cells. With the stem cell pool, the cell itself being able to maintain. So in the blood system, the easiest way to understand the concept of the stem cell is the blood-making stem cell, the hematopoietic stem cell. You have in the bone marrow a certain cell type with a certain phenotype, and that cell, when you climb up on a mountain and you need more red blood cells, as Dr. Rubin mentioned, the hypoxia-inducible factor. When you need more red blood cells, there's different factors that get turned on that cause in the kidney to make erythropoietin, and that tells the stem cell, okay, make more along red blood cells. If you have an infection, you have toll-like receptors being activated, the macrophages, dendritic cells, different cells make GCSF, GM-CSF. The stem cell makes more white cells. You see leukocytosis and so on. Now, the stem cell, the earliest stem cell, the one with the most potential, is the embryonic stem cell. Like the fertilized egg, that's where all the tissues can be made from, which, ironically enough, embryonic stem cells, is a bit of an aside, cause cancer. Right? If you take an embryonic stem cell and you inject it into a mouse without an immune system, the mouse gets cancer. So there's a relationship between stem cells and cancer, which we're going to talk about. All tissues have stem cells. So we know another Canadian discovery, Tillon McCulloch, the concept of the blood-making hematopoietic stem cell. We know that the most well-defined stem cell for the past 40 years has been the blood-making one. In the last 15 years, we found out, when I say we, I mean we as a discipline, we found out the brain has stem cells. In the dentate gyrus area of the brain, there is a compartment of cells that makes progeny cells. When you get old, if you have higher mental function, you have more of these cells. If you are depressed, these cells stop multiplying. In the heart, there are the cardiac-specific stem cells. When you have a heart attack, these cells go into cycle, and they make cardiomyocytes. They don't make enough, obviously, to completely cure the heart, but the body has its own stem cells in different tissues. Injury activates the stem cells to leave quiescence and to make new progeny cells. What are the properties of the stem cells? One, like we talked about, After a heart attack, they come out. I guess the global point I'm trying to make here is stem cells prefer to be quiescent until they're needed. You know, the stem cell's biggest worry is its DNA. Because a differentiated cell, if the DNA becomes mutated, it dies or it gets a proliferative advantage, but it doesn't usually become cancer. It's the stem cell, when that DNA becomes mutated, because it has such potential for self-replication, that's where the cancer happens. So the body inherently wants to protect the stem cells by keeping them, because the more a cell multiplies, the higher potential for the mutations occurs. So the body tries to keep the stem cells in a quiescent state. The body tries to keep them in areas of hypoxia. All the stem cells, from the bone marrow to the brain ones to the cardiac ones, and other ones that we can talk about after, they all prefer to reside further away from the vasculature. Metabolically inactive, and most interestingly, they have drug efflux pumps. The stem cells actually have pumps that when you put on them chemotherapy, they pump this stuff out. You know, when people, I mean, obviously you guys know a lot more about oncology than I do, but for me, one of the things that always, when I started getting into this field, intrigued me was why would the cancer cell all of a sudden start having these multiple drug resistance proteins on them? That selectively you give it the chemo and it pumps it out. The ATP cassette pumps. And then I realized, oh, stem cells have these, and the cancer just uses something that the body has for something else. Now, the other really quick point about the stem cells, there's a lot in the bone marrow. When you administer exogenous stem cells, they accelerate healing. And we'll talk about that in a little bit. Stem cell deficiency is associated with poor healing. So that's normal stem cells. Now let's shift gears here in cancer, and at least one of the reasons why a lot of the people in the pharmaceutical industry are looking back now and saying, why has a quote-unquote war on cancer failed? It's because we were targeting the wrong cell. Tumors, as we heard in the previous lecture, are heterogeneous. They have this stromal component, but in the tumor component itself, you have cells with a variety of different properties. When people develop drugs, what the people do is they take cells from the tumor and they grow them out as cell lines. Those cell lines do not represent the tumor. Those cell lines represent basically an artifact. And this was the big revolution, this knowledge that, let's imagine, for example, that I had colon cancer. If you take my colon cancer and you put it in an immune-deficient mouse, but you put the cells, so you take my cells, you dissociate them, you put them in the mouse, only 0.001% of the cells that I put in for my tumor actually make the new cancer in the mouse. Whereas a lot of the other cells will multiply in vitro very nicely, but they do not initiate the tumor. Because as it was discussed at this symposium before, the tumor needs all sorts of things to establish. Right? It needs immune evasion, it needs angiogenesis, it needs matrix metalloproteases, and so on. The concept here is that the cells that transfer the tumor, those are the stem cells. Those ones are not proliferating highly. And that's a problem. So let's talk a little bit more about how these cancer stem cells are, how we purify them. So you take the tumor, you treat it with different enzymes, and you dissociate it so you have cells at a single level. Then you use methods. Primarily, the main ones have been used as antibody-based. So you have an antibody attached to a magnet, and the antibody recognizes different markers associated with normal stem cells. And the normal stem cells, when you take it out of a tumor, are usually tumor stem cells. So here's one of the markers a lot of people use. It's called CD133. And this is not rocket science, but it's interesting why it took such a long time for us as a field to catch on to this concept. CD133 is a protein that's found on brain stem cells, cardiac stem cells, blood-making stem cells, and so on. And people said, okay, well, why don't we just look at CD133 positive cells from a solid tumor? And you can purify, when you purify them out for the CD133, you can see they make tumors when you put them into mice. So let me actually skip. This is what they look like, CD133 positive fraction versus negative fraction. Even though the CD133 represents a much smaller proportion of the bulk. This is another tumor marker, CD24 negative versus CD24 positive. The first, my God, this seems to be like a Canadian propaganda presentation. This guy, John Dick's also Canadian. I didn't know my friend Brian Sparks here would be in the audience. I think he probably knows. You know John Dick, right? Yeah, from the Ontario Cancer Institute. He's the first guy to actually prove conclusively, long time, 7 years ago, about the cancer stem cell, where he did it in leukemia. So when you make blood, when you take bone marrow, the CD34 positive cells are the ones that contain a lot more stem cells than CD34 negative. So if you look at, this is the number of colonies formed in the mouse spleen. So you take someone who has leukemia, you take their cells, you purify them into CD34 positive, CD34 negative, transplant them in a mouse, kill the mouse, look at the spleen of the mouse, because that's where the leukemia cells make colonies, count the colonies, in vivo repopulation assay, and you can see the CD34 fraction had induced a lot more colonies, and as opposed to CD34 negative. So the stem cell compartment is the one that can transfer the leukemia, the cancer stem cell compartment, because these blasts are not healthy cells, they're all leukemic. So, very quickly to touch on the concept again, what this means. You can't really see this. Okay. This is the cancer stem cell, the one in the middle. Which is a slow multiplying cell. These are the daughter cells, which are fast multiplying. When you come in with the chemo, you actually kill the fast multiplying cells. So the cancer stem cell is not killed, and then it repopulates, and you get the cancer coming back. With other therapies that target the cancer stem cell, you still have the fast multiplying cells, but the fast multiplying cells eventually end up dying, and they don't have the root anymore. So you have cure. That's the vision. The implications of this vision and the supportive data, and we can talk for hours about this, but the implications is more than 8 decades of cell line research, probably more than 8 decades. The first tissue culture was by Alexis Carrel at the turn of the century. So more than 8 decades of research on cell lines. In some ways, we could throw it in the garbage. I mean, well, obviously, we learned a lot from cell lines about molecular pathways, and cell lines are beautiful. But in terms of, I don't know what it's like here, but in California, this month, I guess, is Breast Cancer Awareness Month or something. And every time I go to the store, they're asking me for money for cancer research, for breast cancer research. And I'm like, do you guys know where this money's going? I mean, this money goes to these trials comparing one chemo versus another, which has nothing to do, with the real problems here. So, I don't know. It just bothers me. The reason I don't want to get emotional here, but the reason why it really bothers me is because I got into this field of cancer research when I was young because my mom got leukemia. And my mom's taken ozone therapy. She had very nice results. She's taken intravenous vitamin C numerous times. That's how I met Neil Reardon, with the dendritic cell therapy, placental vaccination, and last month she passed away. And when she passed away, my initial reaction was I threw away all my the papers on leukemia. I said, I hate this field. I suck as a person. Then I started that feeling actually started converting into a feeling of hatred, which was stimulated by these breast cancer people and stimulated by the fact that we know these therapies that are being used by the people in the room today, a lot of them work on this bloody cancer stem cell. And a lot of this concept, like the wound that never heals, and Neil's publication on this years ago, and these concepts that Hugh Reardon started developing, now they're being validated at a molecular level. The acquisition, the $1 billion acquisition of Oncomed, I believe, Oncomed by GSK, $1 billion because they had intellectual property on some of the markers associated with cancer stem cells. These cancer stem cells, I believe, is one of the pathways that intravenous vitamin C, but a lot of these other naturopathic methods are targeting. So, a couple quick points along those lines. Remember how we said in the heart attack that the stem cells start proliferating because they sense the heart is injured. Conceptually, chemotherapy in certain regimens would actually amplify the tumor because the tumor has its mass. It's a three-dimensional mass, which are small tumor stem cells. If you can somehow tell the tumor stem cell not to multiply, try to tell it to differentiate, and at the same time control the fast multiplying cells, you could hypothetically be getting these types of remissions. But on the other hand, if you actively go there and lob off a piece of it with the chemotherapy, that stem cell recognizes, just like the heart recognizes, the heart stem cell recognizes that the tissue around it is injured, there is rationale that chemotherapy in certain situations may make this worse. So that's one of the implications. Now, you're going to say to me, Tom, this is all cool theories, but how can we actually do something with it? Okay, fine, cancer kills you this way instead of this way, but it still kills you. Well, one of the most exciting areas is the differentiation, how to make that cancer stem cell differentiate into a normal cell. And we're going to talk about that. One last point, because I don't know if I covered it in the presentation. The big problem with the cancer stem cells is they're very difficult to culture, to grow outside of the body. So at MIT, there was a publication that came out recently where they've developed some systems. And so the other way to, besides trying to differentiate it, is trying to find specific enzymatic pathways and block those. And the big pharma is doing a lot of this work right now. So again, like we said, how to at least aim to cure cancer. And I know that here at the center, there's a lot of interest in healing and energies. So I put this slide here because I believe there are energies here involved. Neil and Xiaoluang here from the center, they published this idea of cancer as a functional tissue, and like we heard the concept, it's a wound that never heals. If you could heal that wound, accelerate the healing, hypothetically you can cure the cancer. So the concept comes up, what if instead of killing it, we learn to live with it, or we learn it to, we teach the cancer to stop being so aggressive. This concept, who actually first introduced me to, was Judah Folkman, the father of angiogenesis. He believed in this a lot. He believed in choking it, making it die slowly. What we talk about here is trying to make the evil tissue into a good tissue. But how to do this? And one area that we've been working on is using stem cells. So, these healthy stem cells, their propensity is to differentiate into normal cells. For example, an example of this is if you take an embryonic stem cell and you put it in culture, it naturally starts making heart, brain, liver, and so on, just by itself. It has this propensity. So, we're thinking that these cells, because they want to differentiate, they must be shooting off specific chemicals that would tell cells around them to differentiate. We found some work that we didn't do, this Dr. Hendricks et al. Did, where they injected melanoma cells, human melanoma cells, into fertilized eggs. Fertilized egg is undergoing a differentiation process. They saw that the injected melanoma cell would actually start differentiating into non-malignant cells because of the paracrine effect that there's differentiation going on around it. This is not just science fiction. Right now in hematology, the use of differentiation therapy is medical practice in acute promyelocytic leukemia, where you give retinoic acid and the cells differentiate. So the bottom line here is the stem cell is a heal injured tissue. If cancer is a non-healing wound, okay, well, let's give it stem cells and see what happens. That's a fun way to look at it. The bad way of looking at it is this. And Medistem, the company that I work for, we have our own stem cells that were developed at the center, with the center. The endometrial regenerative cells are now we're at the FDA with them doing phase 1 clinical trials in the US. And the reason why this is very relevant is because on the one hand, we're thinking, okay, the stem cells can heal the wound that never heals. But on the other hand, what are we thinking? We're thinking stem cells make a lot of growth factors. Our stem cells have a lot of matrix metalloproteases. They stimulate angiogenesis. Maybe the stem cells are going to make the cancer worse. In any case, we need to know this, and we need to know it fast. So really quickly, the stem cells that we work on is stem cells that we have patents filed on, that were discovered here at the center together with Medistem. They come from the menstrual blood. So basically, this is put into the vagina of the menstruating woman. The women donors are pre-screened. This is all done under IRB, and this has all been approved by the FDA. You take the mononuclear fraction. We have a proprietary cloning process. These cells, the beauty of these is they're a universal donor. So it's a one-size-fits-all, and they get quality-controlled master cell bank. They get put into this little device that then gets shipped off to the doctors at the participating clinical trial sites. Now, that's just to give you a background. These cells, we discovered them here. This is me and Shalong when we won the Publication of the Year award. This is me and my friend from California. This is after we published. It was really good. Different independent groups started publishing the same thing, but we got the patent on them. The cells, like any good stem cell, can become different tissues. Ours make more tissues than others. Ours make a lot of growth factors. They don't cause tumors. They don't accelerate. These initial experiments, it was a UV-induced tumor, and it seemed like they did not accelerate. They actually seemed to inhibit tumor formation. This is the one for slide I want us to focus on, because this one was done in a larger group of animals that was much more quantitative. You take the glioma, it's a C6, it's a rat glioma, and it's blue because it has the gene in it for LAC-Z. That makes it blue when you stain it with beta-gal. And anyway, so the point is you inject in A. This is control. This is the tumors were injected in the brain, and these are cross sections, and this is the overall tumor volume. So we injected the tumors, and then we injected the stem cells. This is intravenous injection of 3 million cells, and this is intratumoral injection. So you can see, as opposed to the control, there seemed to be a reduction in the tumor size. And when we co-stained for the blue, so we know they're tumors, with CD133, we saw that the malignant cells, the malignant stem cells seem to be lower in the mice that received intravenous and intrathecal and intratumoral stem cells. So again, we're not curing here, but we're demonstrating there's not acceleration, but there seems to be an inhibition of the cancer and an inhibition of the cancer stem cell in vivo. So the beauty of this is obviously that our endometrial regenerative cells, they inhibit tumor growth. They seem to selectively home to the tumor site, and they seem to function through reducing the number of tumor stem cells. Now, the next, really quickly, area is another type of stem cell. Is this a universal? And the previous one we published, this one we have not published yet. Cord blood stem cells. They make a lot of growth factors. Dr. Neil Reardon has pioneered the use of using cord blood without destroying the recipient immune system, and has helped literally hundreds of people with this. That was a publication. So the point with the cord blood is you can use the cord blood not just for reconstituting the blood system, but you can use it as a source of growth factors, which is what we've been doing in Panama and treating numerous conditions. So again, the situation with the cord blood is very similar to with the endometrial regenerative cell. Can the growth factors perhaps do something bad? So we started using a system, a very well-defined system, HL60 leukemic cell lines. These are a myelomonocytic cell line that floats. It's an adherent cell line. When it differentiates, if it differentiates in the monocytic lineage, it sticks to the bottom of the plate. So a very simple way of seeing if your intervention is inducing differentiation or not. So here we did a co-culture, percent adherent cells, and added cord blood stem cells, cord blood CD34 cells to the leukemic cells. And we saw a dose-dependent increase in what appeared to be differentiation of the leukemic cells. We also cultured the cord blood cells. We cultured them. We did a C18 extraction. This is a non-specific extraction of a certain molecular weight. And we saw some factors seem to be inducing differentiation secreted by the cord blood. Now, with differentiation, you have inhibition of proliferation. And this is proliferation data where we added the extract from the conditioned media from the cord blood, and we saw dose-dependent inhibition. This is where it became even more interesting. We started treating solid tumors. This is LLC, Lewis lung carcinoma cells that were treated as a lung tumor, a mouse lung tumor. We treated it with these substances made by the stem cell, and there was inhibition of proliferation. This is probably the most exciting one, is where we either injected the products from the CD34 stem cell or the placental matrix, basically the non-defined fraction of a specific molecular weight. We injected them in mice that we gave to the mice an intravenous injection of lung cancer cells. When you administer these cells intravenously in the tail vein, the mice get metastases. We were able to show, using the two different protocols, inhibition of lung metastases using placentally derived factors. And this is tumor size. This is when you actually implant the tumor subcutaneously, and you can watch it decrease. This is control. As you can see, by day 50, the 4 of them died. These are treated ones, and some of them appear to be cured. There was definitely an inhibition of the tumor size, and this is again Lewis lung carcinoma. So the point is, these stem cells, they seem to be making different products that have some anti-cancer activity. Now, Neil Reardon's going to mention, I believe, later tonight some of his more recent clinical data. There's other people such as Mitch Genn, Dr. Mitch Genn. He's also a naturopathic doctor who's been injecting patients with cord blood cells, and he's had some anti-cancer effects, as well as the Chinese group. So there is some support for this concept of the stem cell, the healthy stem cell, inducing a differentiation of the bad cell, of the leukemic cells. Now, what does all of this mean to us here? And this is something where I really would like your input. And I guess the discussion is going to be very interesting for this. It seems to me that the tumor stem cell, the more you can inhibit the inflammation around it, the more you can induce this healing, the more you have a chance of inhibiting the tumor stem cell. But how could we prove this? And how could we prove that some of these healing interventions actually work at the tumor stem cell? So we have the technologies in the mouse, and even in people with biopsy samples, to measure multiplication rate of the tumor stem cells. So I'd be very interested in seeing if there's some ways to combine, for example, in the previous presentation, or this concept of giving the intravenous vitamin C to help modify the stroma, and giving these different factors, I would be very interested in seeing if we can either do some animal studies to examine some of this, or even the possibility of setting up some an extension of some of the ongoing vitamin C studies where we can be getting some samples. So anyway, this is the end of my talk. Thank you.